All right, welcome back, and let's talk about some platelets. Platelets are cool. Platelets are kind of crazy. So, in this chapter, we'll talk about how platelets are super important in clotting, which is very true, but that's actually only 10% of the job of the platelet, although the book makes it sound like that's their only job. And for a really long time, we thought that that was what platelets only did, that they came and they helped clot, and then they left, and that was it. But like I said, platelets actually only have, that's only 10% of their role. They are involved in a lot of other cell signaling and a lot of other cellular mechanisms in order for the rest of the cells to actually maintain regular homeostasis. This is something that I'm actually heavily involved in my day-to-day job, in my full-time job. We do something called platelet-rich plasma injections, and we're doing a lot of studies on trying to figure out how good they are at actually relieving pain and relieving symptoms for patients. So, about right now, the study or the literature says that about half of the patients do improve. Their pain does get or does decrease. And then the other half say they don't even notice a change, which is really strange. So, there's a lot of data that shows that it's pretty 50-50. But there's not a lot of data really explaining why, but it's very clear that platelets have some sort of additional responsibility, not just part of this clotting cascade, which is what we're going to talk about. You will only be required to understand the clotting cascade, so don't worry about understanding the rest of what I just said. I just think it's important to be informed about all of your other options. All right, so platelets, just like red blood cells, just like white blood cells, they have another name. Those are called thrombocytes. Very important in clotting blood. They're replaced every 9 to 12 days. They are recycled by the spleen, and at any given time, about 1/3 are actually preserved in the spleen for any type of reserve. So, if at any point, maybe, God forbid, you got into some sort of accident, you lost an appendage, you're losing a lot of blood, you need a big reserve of platelets at any given time. That's what your spleen is going to help you do. It's going to help provide that extra reserve of platelets, so hopefully you can stop the bleeding a lot quicker rather than just bleeding out. They're responsible for releasing a bunch of chemicals for the clotting process, which we'll talk about. They'll formulate some sort of temporary patch in the walls of the damaged blood vessels, and they will reduce the size of a break in a vessel wall. Whenever we're talking about producing platelets, we're gonna use the term thrombocytopoiesis. So just like we were talking about leukocytopoiesis or erythropoiesis, this is the same thing. We're making thrombocytes. This again also takes place in the red bone marrow, and I highlighted the part of the flow chart where we're talking about this. We start with a myeloid stem cell, we turn into a megakaryocyte, and then eventually we make platelets. 3 things that influence actually making the platelet formation. 2 things. First, one is the thrombopoietin. This is a similar protein to erythropoietin, but this is going to influence the production of platelets. And then interleukin 6, IL-6, and this is going to be a hormone that actually helps stimulate the platelet formation. This will make it a lot more sense whenever you start to actually learn what the other IL hormones and proteins do. I think the last time I looked, it's like there's IL-1 through like the high 20s, and they all have various different functions, but they were all discovered at different times. So I don't know how they're all numbered in whatever order, but IL-6 is one of those main ones that you'll learn about. And then the other thing that will influence platelet formation is really hemostasis, which is the primary clot producing piece. So hemostasis is blood stopping. Hemo, blood referring to blood, stasis, stopping. There's 3 phases of it. First is vascular, then there's platelet, and then there's the coagulation phase, and then finally we actually ruin the clot by getting rid of it. In the vascular phase, we're talking about the vessel itself actually spazzing, and when the vessel spasms, then the diameter will decrease. And when the diameter decreases, then the membranes start to get exposed to different things. The way this was actually discovered was totally by accident. There were some grad students in like the 80s or the 90s, I believe, where they were asked to take some rabbit vessels and throw different types of gases on them for various things, one of which was nitric oxide, which was known to be a vasodilator at the time, and it still is. And when they actually cut the vessels, they actually noticed that the vessels would like spasm and retract on their own. And so they actually started doing additional experiments to figure out how the vessels, like why would the vessels even do that, and they realized it was part of this pathway. So once the vessel gets cut, it spasms, it retracts, it decreases its diameter, and then now we have two different parts of the vessel being exposed to two different environments. So one is the outside of the vessel and one is the inside of the vessel. And the inside of the vessel is going to release chemical factors and local hormones to help recruit certain things. And then the outside of the vessel is also going to wait for other things to be recruited to that area to help stimulate another pathway. Essentially, the thing gets sticky, and then things come to that area. Then we go through the platelet phase, which is where we actually recruit all of these things to the blood vessel itself and create a platelet. And once they finally arrive, then they finally activate to recruit even more platelets. And then once we finally recruit more platelets, then we recruit growth factors. Then we eventually stop those growth factors and tell them, hey, hang on, you're fine, you did your job, just sit tight, stop. Then we formulate a blood clot, and then eventually we coagulate that area where we have enough of a big blood clot that we actually stop the bleeding from the vessel entirely. And then once we're finally done with the coagulation phase, then we need to break down that blood clot through fibrinolysis. So we have an extrinsic phase, which is happening on the outside of the blood vessel, an intrinsic phase, which is happening on the inside of the blood vessel. And then eventually, while both of these phases are happening in tandem, then they converge onto one final common pathway to create that final coagulation or that final platelet plug. So here is where I talk about the extrinsic pathway combined with the intrinsic pathway and the steps of each of those, so become familiar with those. And then finally, those both combine and turn on, the common pathway, where we're finally activating factor X. And then factor X is finally what activates thrombin. Thrombin activates fibrinogen to turn into fibrin, and fibrin finally produces the clot. At the same time, we have this negative feedback control where we have to disintegrate that blood clot. So that's where we have fibrinolysis, where we go and actually break down that clot so that we no longer have the clot. And here are some methods of where we can restrict that process or where we can promote that process, and exactly where this meme from the beginning of the lecture actually comes into play. So where we have all of these platelets getting recruited to the site, we have a platelet party, but at some point fibrin comes in and actually destroys the platelet party, so we no longer have this blood clot because the blood vessel has been repaired, and we no longer need to stop blood from trying to leave because there's no point anymore. That's it for the blood chapter. I'll see you again for chapter 20. Thank you.